This video for Bio 181 for Chapter 7 on cellular respiration is on the second step and the third step in cellular respiration. So cell respiration, we first have glycolysis, and in aerobic cellular respiration, we then go into pyruvate oxidation. So in pyruvate oxidation, we're moving into the mitochondria and we're breaking down pyruvate, which is a 3-carbon molecule, 1, 2, 3 carbons. And we're going to break it down into a 2-carbon molecule called acetyl. So 1, 2 carbons in this molecule called acetyl. And it's called acetyl. It's going to be attached to a coenzyme A, which is why it has this second part to it called acetyl-CoA. We really care about the fact that it's 2 carbons. It's stabilized by the fact that it has coenzyme A. And then we're also going to produce a byproduct over here, carbon dioxide, just has a single carbon. And then this is going to actually diffuse out of the cell. So it's going to diffuse out. So we won't see that again. During this process, we make the byproduct of carbon dioxide, which I mentioned, and an electron carrier, right? So we're making NADH. So again, we're taking electrons and we're going to put them on a molecule called NADH, which is going to take those high energy electrons and bring them to the next step. This is located in what's called the mitochondrial matrix. So the mitochondrial matrix is all of the region in the inside of the mitochondria. So there's a membrane, it's an inner membrane and it's folded, okay, just to increase surface area. So remember structure and function. So it's heavily folded, and this whole region inside of it is called the matrix. So that's where this takes place. So pyruvate was created in the cytosol or cytoplasm, and then it was brought in, and now it's oxidized in the matrix. Okay. If this was a prokaryote, same process can occur. They can have cellular respiration, just occurs in the membrane. Okay, so the overview slide here, we start with the reactant pyruvate. Main idea is that we're going to create a product called acetyl coenzyme A or acetyl-CoA. This is a 2-carbon molecule. We produce also as a byproduct carbon dioxide. This is the first time we're making that. And we produce an electron carrier. Occurs in the mitochondrial matrix. We're oxidizing pyruvate, taking energy out. We take a 3-carbon molecule, we break it into 1 2-carbon molecule, and then another 1-carbon molecule. Okay. So here's the reaction. We start with pyruvate. Here's carbon dioxide being made, diffusing out of the cell. Here is our electron carrier. So 2 electrons hop onto this from pyruvate. Okay. And that's going to be taken to another step down the road. And then we stabilize our molecule with a coenzyme here. So we have this 2-carbon molecule called acetyl coenzyme A. The next step is called the citric acid cycle. So the citric acid cycle also occurs in the matrix. Now we're taking acetyl-CoA, our 2-carbon molecule, and we're breaking it down into 2 carbon dioxide molecules. Excuse me. So we're breaking this 2-carbon molecule, a single 2-carbon molecule, down into 2 carbon dioxide molecules. Here's one carbon dioxide molecule. Here's another carbon dioxide molecule. So we're basically done with the carbon. All of this carbon dioxide is going to diffuse out of the cell. Okay, so we're not going to see it again. We've taken, at this point, all the energy that we can out of the molecule. So in this process, we get another few electron carriers. So we've seen this one before, NADH. And then we're also going to add in FADH2. All right. And we make ATP. So we're making ATP. We're making ATP in this step. And again, substrate level phosphorylation. We're not making a ton. We just make 2 of them. Really, the important part is the electrons that are going to be going with these electron carriers. And they're going to go to the next step. So the summary slide, we start with acetyl coenzyme A. We end with 2 more molecules of carbon dioxide, electron carriers NADH and FADH2, and 2 ATP. This occurs in the mitochondrial matrix. We're oxidizing acetyl coenzyme A, so we're taking energy out. We're creating 2 molecules of carbon dioxide. We're creating electron carriers and a little ATP. So this is the more complex illustration from your book. You do not need to memorize this. It is called a cycle because some of the components are recycled and get used again. Okay, but for our purposes, you don't have to memorize any of that. Just know that we're actually creating the electron carriers here, right? And we're creating carbon dioxide here, right? And we're creating a little ATP here. So the summary of the first steps, glucose completely oxidized. So we've taken all the energy out. We've converted the 6 carbon molecule of glucose to 6 molecules of carbon dioxide, 6 CO2, right? We've made 4 ATP, we've made 10 ADH, and we've made 2 FADH2. The ATP has been used or it continues to be used. The carbon dioxide diffuses out. Here's where the money is, right? This is the key. So most of the energy has been captured in these electron carriers. And so now, whoops, we don't need 2 E's. Now we're going to see where and how we're going to get this energy out. So where's this energy going? That will be in the next video.